So the 4 bases of deoxyribonucleic acid. The purines are adenine and guanine. The pyrimidines, thymine and cytosine, and they form the backbone of the DNA double helix. So an adenine base always pairs with a thymine base, a guanine with a cytosine, and along with a sugar phosphate backbone forms the DNA double helix. These guys became quite famous for this. The DNA, Watson and Crick published their paper in Nature proposing this model of DNA. In 1953, they were awarded the Nobel Prize. Does anybody know when the Nobel Prize in recognition of the magnitude of this discovery? 1962. So it took a long time for the community to realize what this was all about. So, as I said earlier, it's been some time since I've studied meiosis. But in order to have an improved understanding or an understanding of it, what I'd like to do is go through it with you here. So if you want to look at this website, I would encourage you to do that. There's some a very nice animation that goes through the different stages of meiosis and narrates what's going on. You can also stop that and you can do a step-by-step tutorial where there's a kind of like a PowerPoint image and then a description of the phase. And I think if you go there, you'll also find that there are a number of other very interesting presentations. There's one on mitosis, so you can compare meiosis to mitosis. If you're interested in the independent assortment of alleles or Mendel's law is Mendel's experiments, the Meselson-Stahl experiment, which was an experiment to essentially confirm that what Watson and Crick said was correct. High throughput sequences, and there is a lot more. And as I've searched for a really nice animation of meiosis, this is the website that I found that really is something that I would encourage you to look at if you're interested in a real understanding and ability to stop the presentation, go back, study it again, and so on. This is just one of the images that was on that website. And so it describes meiosis, the 2 stages of meiosis, meiosis 1, meiosis 2, and it describes each of them. And there's also another one that's the same thing but in a different format. And I think it's really quite nice. And so these images will be in your package, but if you want to study it again, that website I think is a very good place to spend some time. So I've taken those steps and I've just put them into this chart here. And the intention is to make sure that we understand in particular some of these phases that have more to do with synapsis and crossing over in particular. So meiosis begins with a microspore mother cell, a diploid cell, and it begins meiosis 1 in the prophase stage, where homologous chromosomes begin to pair, synapsis occurs, and crossing over occurs. Those are the events that decide on how genes are transmitted, which genes are transmitted and in which ratios. Metaphase, in the metaphase stage of meiosis 1, the homologous chromosomes are paired and form bivalents. Each of those bivalents consists of 2 chromatids. Then anaphase 1, the homologous pairs separate and move to opposite poles of the cell. Telophase 1, a nuclear membrane forms around those separated bivalents. And at the end of telophase 1, we have cytokinesis 1, when each of the 2 cells now has the same number of chromatids as the parent cell. So we've gone from 1 cell that's diploid to 2 cells that are diploid. And then the second phase of meiosis begins with prophase 2. In prophase 2, the chromosomes are composed of 2 chromatids. The single chromosome, in metaphase 2, the single chromosomes are at the equatorial plate of each cell. In anaphase 2, the single chromosomes, each with 2 chromatids, separate and begin moving to opposite poles of the cell. And then in telophase 2, cell membranes begin to constrict and form 4 separate cells, so that at the end of cytokinesis, we now have 4 haploid germ cells, with each of them with a different genetic constitution based on what happened up here in prophase 1 with synapsis and crossing over. So what I'd like to do now is go and look a little closer at prophase 1. How the homologs pair, what happens with synapsis, and what happens with crossing over.